Thank you everyone for listening to this short presentation. I want to briefly touch base about Seer's Proteograph technology, a multi-nanoparticle approach for identification of novel biomarkers in Alzheimer's disease. On the left, I'm showing the proteome coverage across a cohort of 200 samples, 100 Alzheimer patients and 100 normal people. And we did two different mass spec acquisition mode, DDA and DIA. And for DDA across the cohort, we are getting close to 5,000 protein groups. And on average per patient, we are getting about 2,500 protein groups. With DIA, we are getting a bit better number, over 5,000 protein groups across the cohort, and per biosample on average over 3,000 protein groups. Very good numbers. In terms of classification performance, we try to separate Alzheimer from normal people, and this is a hard job specifically for Alzheimer's disease because the signal is quite weak in plasma. Knowing that, after tenfold cross validation, we got a 75% area under care for RSE, which is considered a notable performance for Alzheimer's disease in plasma. Doing pathway enrichment on top features coming from this classifier, we get relevant Alzheimer pathways such as complement and coagulation cascade, IGF, and there are a lot of research supporting this pathway for Alzheimer, showing here in red and green. We also identified novel biomarkers that we didn't put here, and happy to provide more information later on. For the pQTL analysis, we looked at the SNPs coming from the genome, whole genome array, and their association to the changes in protein expression. And this should enable better identification of causal protein expression and causal SNPs. And for such a small cohort, we identified for DIA about 60 and for DDA about 76 pQTLs, corresponding to 20 protein groups. These are very notable numbers. Again, for such a small cohort, we made sure the FDR are quite conservative for these protein groups, and we found notable Alzheimer biology within these 20 protein groups. In overall, we believe the Proteograph technology enables deep and unbiased discovery of proteomics at a scale. Thanks for listening.